Good afternoon. Welcome to our first seminar of the semester, the Muscle Health Research Center. My name is David Hood. It's a pleasure for me to introduce our speaker today, Dr. Nadine Wiper-Bergeron. Dr. Wiper-Bergeron is originally from the city of Ottawa. She grew up there, and then she went to McGill University for her undergraduate degree in biochemistry. And then she moved to Ottawa, back again, to fast track into the PhD program. No master's degree, just zipped right into the PhD program in biochemistry. And she was supervised by Robert Hachey. That name may ring a bell to some of you. And when she finished her PhD, she did not do a postdoc, but was hired right away as an assistant professor. And now she's also a full professor at the University of Ottawa in biochemistry, in cellular and molecular medicine, and also an assistant or associate dean. So she's risen through the ranks rapidly, Dr. Wiper-Bergeron has. So it's a pleasure to have her here today. She's going to talk about linking inflammation to muscle wasting in cachexia, a role for CBP beta in tumors and muscle. So Nadine, please turn on your camera and your microphone and share your slides with us. We're anxious to hear about this very interesting topic.
Wonderful. Thank you so much. I'm just going to get the share started. Here we go. So thank you so much for the invitation. I'm really happy to be here and to share with you some of the work that we've been doing. It's really wonderful. The muscle community is such a great community, and I recognize it's Friday afternoon and you are 45 minutes away from TGIF, but I really appreciate the fact that you're here with me this afternoon. So the topic that I'm going to talk about today is a sort of a major project in the lab that we've been working on, which is related to the mechanisms that are underlying the development of muscle wasting in the context of cancer. And I'll fully admit it's an extremely complicated model to work with, and I think that's what makes it really exciting and at the same time somewhat complex. So I'm hoping that I've made the story streamlined enough. So what my lab is interested in is sort of the transcriptional pathways that regulate adult stem cell differentiation and quiescence. And really my interest started in this sort of mesenchymal stem cell, this idea that there were stem cells that were present, especially in white adipose depots and in the bone marrow, that had this sort of multipotent potential to develop into fat and bone and muscle. And certainly my PhD work was really largely concentrated on adipogenesis and sort of the mechanisms, the transcriptional mechanisms regulating that. And in that time, it's where I started working with this particular transcription factor called CAT enhancer binding protein, which I'll introduce more completely shortly. And so a lot of my work was related to this, and CBP beta is a very, very important factor in terms of promoting adipogenesis. And as I developed my own lab, I started looking at what the role of this factor was in sort of lineage decisions overall. And this led to some discoveries that CBP beta was actually quite inhibitory for the bone lineage, but more importantly, sort of a serendipitous discovery that it's actually quite inhibitory also for the muscle lineage. And this particular finding was quite novel. There was really nothing in the literature about this transcription factor in the muscle lineage. And so it became sort of everything for me. There was just so much that we could discover in that context. And so we've done a lot of work, which I'll introduce some of the basic stuff, but I'll mostly focus on all the work that we've been doing in the context of cancer cachexia and how it relates to inflammation, and maybe tease you with some unpublished work that we've been doing. So what is cachexia? Well, cachexia is a wasting syndrome. It can be present with multiple different conditions, including things like rheumatoid arthritis, COPD, AIDS, but the model that we're working with is cancer cachexia. And so this is a paraneoplastic syndrome that affects up to 80% of cancer patients. It is characterized by a massive loss of skeletal muscle, and systemic inflammation. And those are really the only two criteria that we use to develop it. It seems to be the cause or the direct cause of up to 30% of cancer deaths. So it leads to a really important frailty. And because of this, it does make patients less able to withstand the challenges of anti-cancer treatments, and it can reduce the therapeutic window as well, where we can effectively treat tumors themselves. So I see cancer cachexia as a real burden because it does actually make it more challenging. It reduces quality of life, but it also makes it more challenging for us to treat the tumors themselves and therefore extend patients' lives from the get-go. So I think it is a really, really important health problem. Now, the weight loss that you see in cancer cachexia often involves wasting of the fat mass as well, but cachexia can occur in individuals with obesity. It's often also paired with anorexia and malabsorption, so changes in the gut itself, and these can actually exacerbate some of the wasting. And certainly in cancers of the GI tract, we can definitely see those things being involved. That being said, nutritional support, direct nutritional support, doesn't reverse cachexia. So the wasting that we're seeing is independent of the ability to absorb and use the nutrients that we're bringing in. So it is a multi-organ, very complex paraneoplastic syndrome, but at its core, it's systemic inflammation. Now, the question of where that systemic inflammation comes from is one that the field hasn't really adequately answered. So it's long been sort of quoted, and if you look at the literature, they will say, well, it is the, inflammation produced by the tumor or the host response to the tumor that is responsible for this systemic inflammation. And I think what I'm hoping to convince you of today is that it's probably a little bit more complex than that. And to be honest, it's always kind of bugged me that tumors can be quite small but cause massive muscle wasting. And so to me, there was always a scale issue that made me think that there was probably multiple steps between the tumor and the muscle wasting itself, and that to truly understand or to be able to treat or to be able to stop cancer cachexia from developing, we have to have a much better understanding of those communication pathways between the tumors and these different organs as well as the muscle itself. So obviously, cachexia is definitely a multi-organ syndrome, and there's dysfunction that has been identified in various organs in the context of cachexia, and all of these can directly impact skeletal muscle health. So, for example, as I mentioned, anorexia or changes at that level, sickness, behaviors, reduced appetite can obviously promote muscle wasting because we're not bringing in enough nutrients. There seems to be some evidence that this systemic inflammation promotes browning of white adipose tissue, and so increasing thermogenesis and therefore also creating a negative energy balance. So all of these things would be expected to have a negative impact on skeletal muscle itself. They're not necessarily muscle intrinsic, but they do impact on muscle health. But there are nonetheless muscle intrinsic changes that happen as well in response to the tumors. And these we can model in culture, and we can certainly see them in vivo as well, both in mouse models and of course in human patients with cachexia. And so these changes that are muscle intrinsic are also expected to contribute to the dysfunction of these other organs. And so you can see right away it becomes sort of a bit of a complicated mess because the muscle itself, as it changes, can provide and secrete cytokines and myokines and other substances, and given its overall mass, can have a big impact on other organs around. And so there's sort of what's demonstrated in this image here, sort of an interplay between various organs that are working towards bringing nutrients and are also using energy, and where that energy is coming from the muscle, and then how the muscle actually affects those as well. So you can imagine when you get into an animal model or you're looking at samples from patients, all of these things have to be accounted for. And so it's a quite complicated model to work in, but one that is important. So what we've done is to look at specifically the interplay between the tumor and the muscle in somewhat reductionist models to allow us to eliminate some of the changes that might happen with appetite or browning and other sort of overall metabolic changes that make the interpretation of the data a bit more complicated. And our primary focus is understanding what's happening at the level of the muscle itself. Okay. So in response to tumors, we know that several signaling pathways can be activated. The sort of central idea was that these tumors were secreting, or the host response to the tumor itself was causing the secretion of inflammatory cytokines that stimulate signaling pathways such as NF-kappa B. So these would be things like tumor necrosis factor alpha, IL-1 beta, and IL-6, which have been long described as the pro-inflammatory cytokines in the context of systemic inflammation that are bad for muscle. And these pro-inflammatory cytokines can certainly stimulate the expression of ubiquitin ligases in myofibers that promote directly the degradation of sarcomeric proteins. And so there's a certain catabolism of proteins that exist in myofibers leading to their atrophy. But there's also evidence that these signaling pathways that get upregulated can also interfere with signaling pathways that promote hypertrophy, and so that we're not getting any anabolic response, and potentially also regeneration. And so that's something that I certainly want to discuss in more detail. So, this sort of overall central hypothesis for cachexia, that you have this sort of intrinsic muscle wasting, that you have upregulation of these ubiquitin ligases that lead to degradation at the level of the sarcomeric protein, has led to many efforts to try and identify tumor-derived proteins that drive this wasting, right? So what are the common factors and all that these tumors are secreting that lead to the upregulation of MRF1 and atrogen-1, and therefore muscle atrophy? And to be quite frank, I mean, it's been quite difficult to identify one single factor or a group of factors that are common in all human cancers that lead to cachexia. So not only is there a lot of variability between the different types of cancers that are associated with cachexia, not all patients will develop cachexia to the same extent, and certainly every pancreatic tumor is a little different as well. Every lung cancer is a little different as well. So there's so much variability that it's been very challenging for the field to sort of identify one druggable target that we can block and protect muscle. And the field certainly got more complicated when we realized that in fact, in the context of cachexia, there's actually damage that is occurring to the muscle. So it's not just the upregulation of signaling pathways that lead to degradation of sarcomeric proteins inside fibers, making them smaller, but there's actually membrane level damage as well. And so this image is actually from a paper from Dennis Guttridge's lab, and they looked at mouse models, but also human patients with pancreatic cancer. And as so this is a patient with no cachexia. In this row here, it is a patient who's got about 10% weight loss, and here 22% weight loss. And what you can see is with staining with IgG, but also in the laminin staining, that the condition of the muscle is actually degrading. So this was a really important discovery for the field to understand that this is not only about signaling that is causing shrinking of the fibers, but the fibers are actually damaged. Because once you get fiber damage, you immediately ask yourself, well, is it being repaired, right? So in other models of wasting, like muscular dystrophy, for example, if we get myofiber damage, we expect there to be sort of an intrinsic repair process that comes after that. So certainly this group probably doesn't need any introduction to muscle regeneration at all, but just in case. So of course, there is normally, in response to myofiber injury, the muscle resident adult stem cell population called satellite cells will become activated. They will proliferate and differentiate. These cells will then fuse and be able to form new myofibers that will repair the injury itself. So in the context of cachexia, we would of course expect that if there's myofiber damage, that would lead to a regenerative response, as we see following acute injury with compounds like cardiotoxin, for example. So obviously this led to the very simple question of, is muscle regeneration happening in the context of cachexia? And so this is data that's actually coming out of my lab, and I'm just showing you one of the models that we use to study cachexia. And so this is a tumor inoculation model using a cell line called Lewis lung carcinoma, which is a spontaneous lung tumor that was developed in the C57 black background. And so you can take these cells and culture them and inject them subcutaneously into the mice, leave these mice for 3 to 4 weeks. They will develop, of course, a tumor and also wasting. Now, it's a relatively mild cachexia, and that we're talking, somewhere around, 20% muscle loss, but it's actually very, very reproducible. And so I'm just showing you some of the metrics here of the overall weight differential between sham injected mice and tumor bearing mice. Their food consumption doesn't change at all. So at least for this particular model, one of the reasons I like it is that it doesn't interfere with the mobility of the mouse or its food consumption. And so we can attribute changes to the muscle quality to, in fact, the tumor itself. So here on panel C, what I'm just showing you is that, of course, when they're tumor bearing, we have a loss of the TA muscle weight, and I have some cross sections here of the TA, just showing you about a 20 to 25% loss in the overall cross sectional area. So one of the things that's notably absent from this cross-sectional area is the presence of centrally located nuclei, which is, of course, what we use to identify newly regenerated fiber, which suggested to us that, in fact, regeneration was not occurring in the cachectic mice, despite the fact that muscle injury was, in fact, happening. So to test this, we actually sort of used our cachexia model, but layered on an acute injury to be able to really stimulate muscle regeneration. And so the acute injury model that we used was cardiotoxin, but we also did this with barium chloride. And so what we did is injected the mice with the LLC cells to inoculate the tumor, allowed the tumor to grow for 3 weeks. So these mice had started to develop cachexia, so it was a very mild but early cachexia. Then we did a cardiotoxin injury to the left TA muscle. We left the right TA uninjured, allowed these mice to recover for 1 week, and then looked at repair as you normally would for this kind of injury model. And so what I'm showing you here is hematoxylin and eosin staining of the sham mice before and after cardiotoxin injury. And so these are, in fact, the black bars shown here. This is the TA weight. So you can see that, of course, the TA after cardiotoxin injury has almost entirely restored its cross-sectional area at the end of the experiment. For the tumor-bearing mice indicated here by the LLC, well, these mice are already cachectic, and so in the absence of injury, the contralateral uninjured leg, we can see already that there's a drop of about 25% in terms of muscle mass. And for the injured leg, we can see that, in fact, we have a further reduction of about another 25% in these animals. But I think the really telling information is to look at the hematoxylin and eosin staining, where we can see that the muscle condition is really terrible. And so, yes, there are myofibers that are regenerating, but not only are they quite small, but they're actually quite rare. So there's really a lot of infiltration still in this muscle. So it really doesn't repair well. So this really told us that, in fact, muscle regeneration is impaired in cachectic animals. And that this may, at the time that we were doing these experiments, we didn't have as good information as we do now about how important the inflammatory context in muscle is for directing stem cell activities and also repair itself. And so I think we'll be able to put this data in context as I move forward. Okay, I know it looks like my pointer is freaking out, but it's because I can't seem to advance my slide. Okay, so obviously the next thing that we wanted to do was to understand, well, okay, if regeneration isn't occurring, has something happened to that satellite cell population, to those stem cells that are resident in the muscle? We know that they're responsible for the reparative response. So the first thing we did was to look at that an uninjured cachectic muscle and say, well, just quantify the number of PAX7 positive cells or satellite cells that were actually present in TA sections in sort of healthy animals versus cachectic animals. And so what you can see here, and this is just the immunofluorescence pointing out the satellite cells, is that the actual number or the percentage of satellite cells increase significantly in the cachectic muscle. So they're not repairing injury, but the satellite cell numbers are actually quite high. And this is, independent of any changes in overall number of nuclei. If we look at the injured model that I just showed you, where we take a cachectic animal and we actually injure them with cardiotoxin, again, this is just a repeat of the TA weight just to show you what it looks like overall. But what you can see is that, of course, in the sham animals, so in black, the number of PAX7 cells expands a little bit after injury, which is expected, and it's what we see. But this is in fact increased both in the contralateral uninjured leg and expanded significantly in the injured leg. So what we have is no repair. We have muscle wasting, but we have a lot of PAX7 positive cells that are there. So it seems like this satellite cell population is able to expand in the context of cachexia, but they don't really efficiently contribute to repair at all. So they're sort of stuck part way where they seem to have gotten activated, but don't transition into differentiation. So as a consequence, you get poor muscle regeneration, and this would be expected to exacerbate wasting. So we wanted to understand why regeneration was blocked in this model. So what I'm showing you here is just, again, sort of that time course of the satellite cells on an uninjured muscle fiber. When there's injury, these cells will activate and proliferate. Some of them will certainly self-renew, move on to differentiation, fuse, and repair this muscle. So what we had been doing, and as I mentioned earlier, my lab had been studying the role of a transcription factor called cat enhancer binding protein. This is a member of the BZIP family of transcription factors. So it's a basic leucine zipper that allows these factors to homo and heterodimerize and to bind DNA. So it has fairly broad tissue expression, and in fact, it's probably one of the most common, its binding site in DNA is probably one of the most common response elements in mammalian promoters within the first sort of 300 base pairs of the transcriptional start site. So it has broad expression, but it seems to have very specific roles in the body. So there is a knockout mouse that was created for cat enhancer binding protein, and the complete knockout mouse had very, very high perinatal mortality. And this was due to an absence of white adipose tissue, which is sort of my old work, defects in the formation of brown adipose tissue, and profound immunodeficiency, among other developmental issues. So there were no obvious skeletal muscle defects, but there was certainly problems with immunodeficiency, which became interesting to us later. Now, as I was saying, as I was looking at sort of these lineage decisions from mesenchymal stem cells into fat and bone and muscle, we found that, in fact, expression of CBP beta in that model completely blocked myogenic differentiation. So there was absolutely no myogenic differentiation that occurred, which spurred us to look to see what the role of CBP beta would be in muscle, and in particular in the satellite cell population. So what we found is that in healthy muscle, CBP beta expression is really found in the satellite cell and not in the myofiber at all, at least not in healthy muscle. And its expression was highest in quiescent muscle cells, and this was absolutely critical, we found, for establishing quiescence and for allowing for satellite cell self-renewal and return to quiescence. And I've just placed the papers here if you're interested in seeing them. And so that expression then decreases sort of in parallel to PAX7 expression as these cells progress through activation and differentiation. In fact, we found that CBP beta actually is upstream of PAX7 and regulates PAX7 expression in this adult stem cell population. So as CBP beta levels decrease, MyoD levels increase, and the cells are proliferating and eventually will differentiate. And so what we know is that the CBP beta levels are actually quite low in fully differentiated cells. So, if you force expression of CBP beta in myoblasts through overexpression, as I've done here, these are in primary myoblasts, but it's true in C2, C12 as well. What you get is, and so I'm just showing you here the expression of CBP beta, it forms 3 isoforms. There's only really one that seems to be predominantly expressed in muscle, and this is this isoform called LAP. It was initially described as liver activating protein. So it's an activating transcription factor isoform. So every time you see LAP, that's just basically the CBP beta function isoform that is present in muscle. So you can see here, this is just to confirm the overexpression of CBP beta in these myoblasts. And when you do this and you compare it to sort of the empty vector controls, what you find is that the PAX7 levels stay high, and the myogenic regulatory factors and as well myosin heavy chain expression actually decrease. And so what this looks like is that the cells really don't differentiate very well at all. So very, very few cells become myosin heavy chain positive, and so the differentiation index, or the percent differentiation, is significantly decreased when CBP beta expression is maintained in these cells. And of the cells who do form myotubes or do become myosin heavy chain positive, they're actually very, very small, as indicated by the fusion index. And so for me, the fusion index is the number of myonuclei per myotube, whereas the differentiation index is the percentage of the nuclei that are found in a myosin heavy chain positive cell over the total number of cells in the dish. So the differentiation index gives you an idea of how many of the cells or what percentage of the cells differentiated, whereas the fusion index gives you some measure of the size themselves. So there's poor differentiation when you overexpress CBP beta, but as well, those that do manage to differentiate are very, very small. Okay, so if we get back to cachexia then, I mean, obviously the opposite is true. If you knock out beta, they precociously differentiate. They are somewhat resistant to growth medium and the factors in them. They make bigger myofibers in vivo and so on. But if we were to get back to cachexia, you can actually sort of model muscle wasting in culture by incubating myoblasts or fully differentiated myotubes with conditioned media that comes from tumors. So what I'm showing you here in panel G is actually a panel of a bunch of different tumor models that you can use to study cachexia in culture, and it's only a subgroup of them. There are many, many more. And so what is shown here is actually we took the conditioned media from these different tumor models and actually put them on myoblasts and looked at CBP beta expression. And so what we can see is that the conditioned media from multiple different tumor models can actually induce CBP beta expression in myoblasts. And what we know is when we induce CBP beta expression in myoblasts, we block differentiation. And so what I'm showing you here is this in culture experiment where we've actually taken proliferating myoblasts and co-cultured them or cultured them with conditioned media from 2 different tumors, the SKOV, which is an ovarian tumor, which is shown here, which doesn't increase CBP beta levels as high as other models, versus PC3, which is a prostate cancer cell line that actually increases CBP beta levels more. And so we compared the 2 of them in terms of their ability to interfere with myogenic differentiation in this model. So the cells are incubated for 2 days in 50% conditioned media, either from this SKOV3 cell line or the PC3. And after that, they're transitioned into differentiation medium. And what you can see is that the SKOV3 cells do differentiate, and this is a myosin heavy chain stain that you're seeing, whereas the PC3 are a little bit inhibited in terms of their differentiation. And so this is obviously quantified again in the differentiation index, so a reduction in differentiation when they're incubated with tumors that lead to higher CBP beta expression in the myoblasts, and also a decrease in fusion. So obviously, if the cells aren't differentiating well, then there's going to be more PAX7 cells that are left in the dish, and that is in fact true, which looks a lot like what we see in vivo. In fact, if we go back to the in vivo cachexia model, what we see is, of course, yes, this increase in PAX7 numbers, but also an increase in CBP beta expression as well. And so not only are there more PAX7 positive cells, but these PAX7 positive cells are also strongly CBP beta positive. So this kind of led to the hypothesis that, of course, CBP beta expression in these in the muscle satellite cells in the context of cachexia was what was in fact preventing myogenic differentiation from happening. And so, yes, there was pressure that was causing wasting in the myofibers, but we were not actually able to elicit a regenerative repair, which would be able to sort of offset some of the damage that was happening with cachexia. Okay. So to try and understand how CBP beta blocks myogenesis, we performed a bulk RNA-seq on C2C12 myoblasts that were retrovirally transduced to either overexpress CBP beta or with an empty virus, and so that's what I'm calling PLX here. And this was done both in growth conditions and also in differentiation conditions. And so what we did is looked at the differentially expressed genes that we found with CBP beta expression, and did, of course, the gene ontology analysis of the differentially expressed genes to see sort of what were the top terms that were coming out. And what I'm showing you actually in panel B here is that if you actually look at the top terms, they're all really highly related. So glycoprotein, signal peptide, extracellular region, secreted, extracellular matrix, heparin binding. So when we looked at this, we realized, wow, most of what is being differentially expressed when we overexpress CBP beta in myoblasts are in fact secreted proteins, right? They're part of a secretome. That in fact it is, CBP beta seems to be directing the expression of a particular secretome. So we next co-cultured CBP beta overexpressing myoblasts with wild type myoblasts, which I'm showing you here, and looked to see whether or not these cells could differentiate normally. And what we found was that, in fact, the secretome from a CBP beta expressing myoblast was actually sufficient to inhibit differentiation and fusion in a wild type cell. So it really did suggest that it wasn't necessarily CBP beta expression inside the cell that was changing gene expression within the cell that was changing its ability to differentiate, but rather that it was producing a secretome, and that secretome could act in either an autocrine or paracrine manner to interfere with differentiation and potentially myotube size as well. And so what I've included here in panel H and panel I are in fact myotube diameters. So we can see that not only are is fusion inhibited and differentiation is inhibited, but the myotube diameter and the area that is covered by myosin heavy chain positive cells is also reduced. So to understand what these components of this CBP beta dependent secretome was and what was in there that could potentially be the cause of the inhibition of myogenic differentiation, we sort of classified this secretome further and looked for sort of overrepresented groups. And what we found overrepresented were in fact cytokines and chemokines. And so what I'm showing you here in panel C is actually a collection of cytokines and chemokines that are overexpressed when CBP beta is expressed in muscle satellite cells. So there were also several growth factors that are known to inhibit differentiation that were also produced in CBP beta overexpressing satellite cells, and so that also can certainly contribute to the inhibition of myogenic differentiation that we see in the context of cachexia. So in this list here, I mean, there are several factors, including IL-1 beta here, which is a known cachexin, so a known pro-inflammatory cytokine that causes damage to muscle. In fact, all of these factors here that are indicated in red are not expressed at all in normal myoblasts and become expressed when CBP beta is present. So this kind of goes back to when I introduced CBP beta and the knockout mouse, and I said, well, they have profound immunodeficiency. As it turns out, CBP beta is a really important regulator of macrophage function itself, and seems to play a very important role in sort of pro-inflammatory processes, including the acute phase response in the liver, but also in the production of a large number of cytokines. And so the more we look at this, the more we're shocked at how many of the targets that we can come up with for CBP beta that are actually cytokines, and what proportion. So it's like 80% of the most cytokines are regulated by CBP beta expression. So we really think that, you know, from this point, we were really considering that CBP beta could be sort of a central regulator of this sort of pro-inflammatory secretome, and that this could be really key to understanding what was happening in the context of muscle. So this is just sort of a graphical way of explaining this. So we have, of course, the tumor. We know the tumor secretome is producing all kinds of factors that are reaching the muscle, and that this is driving CBP beta expression in the muscle satellite cells, which is changing them. And what we predict is that it's changing them to produce a secretome that is now can act in an autocrine and paracrine manner, to interfere with myogenic differentiation, and potentially, as I said, IL-1 beta is produced to affect the myofiber health next to it. And this would, of course, lead to atrophy, both by degrading proteins within the myofibers, but also by suppressing the regenerative response, overall leading to progressive loss of muscle mass. So what I've included here is actually a Western, just sort of pointing to this secretome coming from the tumor, since we don't have a lot of detail about what that is. And it is actually quite variable depending on which tumor and what tumor model you use. But I'm just including here a Western blot looking at the effect of sort of those top three pro-inflammatory cytokines, so the IL-1 beta, IL-6, and TNF alpha. And all of these, when we treat myoblasts with them, can upregulate CBP beta. So CBP beta does seem to be very, very sensitive as well to pro-inflammatory signaling. In fact, it was initially described as the transcription factor responsible for IL-6 signaling. So it's really always been part of this loop. It seems to be upstream because it is producing a whole bunch of cytokines, but it's also downstream of cytokine signaling. So the water is getting a little muddy, but it opens the possibility that the tumor may produce pro-inflammatory cytokines, and the action of that might be to amplify the inflammatory signal in target cells. And so cells that can respond to that secretome can then perhaps themselves upregulate pro-inflammatory transcription factors, including CBP beta, and this leads to the production of a secretome that is also procachectic. And so it allows us to have a relatively small tumor, but a signal that gets amplified in muscle, which is much larger. And in doing so, and in creating this sort of inflammatory environment in muscle, we have the potential to actually create systemic inflammation and therefore affect multiple organs. So we really, hypothesized that, if CBP beta was critical for the development of a pro-inflammatory secretome in muscle and by muscle satellite cells, and that this was really foundational for inflammation, then maybe CBP beta expression in the tumor could do the same thing. So it was a bit of a leap, to sort of go back and go, well, if CBP beta seems to be so important in regulating cytokines, and if it is so key to producing this secretome, it's such a central regulator of the secretome, then maybe its expression in tumors can help us distinguish between tumors that produce cachexia and those that don't. So we kind of looked at the literature a little bit and available data sets that were around to see whether or not this idea had any weight at all. And so looking at colorectal cancer, so each of these dots are samples from colorectal cancer patients. And this is just using publicly available data, comparing healthy tissue adjacent to the tumor tissue and tumor tissue, what you can see is that the CBP beta expression values increase significantly in the tumor versus the healthy tissue. If you actually look at the TISH database, so this is a single cell RNA-seq database looking at different tumors. So these are all different types of human cancers and sort of what functions they're most highly correlated with. What you'll notice is if we inquire for CBP beta, right in the middle here, it is always correlated with inflammation. And so this led us to look more in detail, even in the tumor microenvironment, and looking at cancers that were very highly associated with cachexia, including pancreatic cancer and non-small cell lung cancers. What you see is CBP beta expression is actually quite high in the malignant cells, as well as other cells in the microenvironment, which is maybe a story for another day. So, this suggested that maybe high levels in the tumor could actually promote a pro-inflammatory secretome, and then that would be sort of the initiating event leading to muscle wasting in the context of cachexia. So to test this sort of directly, as I said, we went back to our more reductionist models of cachexia, and so we did this in culture first, and we took those tumor cells, the LLC cells that I showed you that we use in vivo, and we took these cells and we actually just knocked down CBP beta expression using a lentiviral vector, SH-mediated lentiviral vector. And then what we did is we redid the in-culture experiment. In this case, we really did an atrophy experiment. So the myotubes were fully differentiated for 4 days. And then after that, we added conditioned media from the LLC cells with the empty virus infections, so our controls, or those that had CBP beta and knockdown, and that's just the validation that I'm showing you here by Western. And so what you can see in the immunofluorescence here for myosin heavy chain, these are what the cells look like on day 4, so at the time of the treatment. If we leave them in unconditioned media, certainly you can see that in the 2 days that happen afterwards, there's still an increase in myofiber size and differentiation. So they hadn't completely finished differentiation, which I think confounds the analysis a little bit. If you include the LLC cells with the control SH vector, well, here we see the atrophy again, and that's actually quantified here in the blue. So if you compare the gray bars, which are the unconditioned medium here, to the blue bars, which is the SH controls, you can see that, of course, we have a decrease in myotube diameter, a decrease in the area that's covered by myosin heavy chain, a decrease in the fusion index, and of course, in differentiation. Now, if you knock out CBP beta in the tumor cells and you put conditioned media from those cells on, you actually protect myotube diameter, the myosin heavy chain positive area, the fusion index, and the differentiation index. So basically, these cells are no longer able to reduce myotube size. We then did the opposite experiment, and we actually took a lymphoma tumor line called EL4, which you can also use in vivo in the C57 Black 6 model, and we redid exactly the same experiment, except now this tumor actually doesn't cause cachexia very much, and so it's actually now it's the blue bars here, so it really doesn't cause any decrease in myotube diameter and has very little impact on the fusion index, no impact on differentiation index. So in these cells, what we did is we overexpressed CBP beta. So we forced them to express higher levels of CBP beta, which is shown here in the Western. And when you do this, we get wasting. So the myofiber diameter decreases, the myosin heavy chain positive area decreases, the fusion index decreases, as does differentiation. So basically, just playing with the levels of CBP beta in the tumor determines whether or not that tumor will cause wasting or not. Obviously, we repeated this in vivo, and so we took those same cell lines from the LLC model, the SH control, and the ones that have the beta knockdown, and we actually made tumors with them. In panel B, I'm showing you TA muscle and just showing you the relative decrease in cross-sectional area in the SH controls because we have cachexia in that model, and the protection when we use a tumor that has SH beta. This had nothing to do with tumor size. Regardless of tumor size, the SH control tumors always generated more muscle wasting overall. And so that's just quantified here in terms of the overall cross-sectional area. And of course, you can see it here in a frequency graph as well, that the both the sham, so the healthy animal cross-sectional areas, and those from the tumor-bearing animals that had CBP beta knocked out are more or less equivalent. So we basically protected them from cachexia, and this is just a binning of small, medium, and large fibers, just to show that, of course, there are much more small fibers in the SH control tumors, and this is actually reversed when you knock out CBP beta. So obviously the CBP beta expression in the LLC cells was critical for these tumors to be able to induce cachexia. We did this with the EL4 model as well. So the EL4 model normally does not cause cachexia, and so it's the gray line here, and the black line is the sham. What's shown in blue are actually the fibers from the TA muscle, the cross-sectional areas from the TA muscle in animals that have tumors that are overexpressing CBP beta. So it's quite the opposite again. Now we overexpress beta in a cancer, in a tumor that does not cause cachexia normally, and we induce wasting. So this really suggested that CBP beta expression in the tumor was both necessary and sufficient to be able to generate a secretome that causes wasting, both in a reductionist model in culture, but also in vivo. So to understand what in the CBP beta dependent secretome is promoting wasting, we did some cytokine profiling and compared the secretomes of CBP beta expressing myoblasts to wild type myoblasts, and also LLC cells, compared them to the ones that were knocked down for CBP beta expression. And so that's just what I'm showing you here, is these little dot blots that come out of the cytokine profiling. So it's a little bit of a biased way of doing it, and that we're looking at about 100 different cytokines, but we're definitely focusing in on cytokines. These are the heat maps just showing the relative expression of cytokines that go up when we express CBP beta. And of course, in the tumor cells where we've knocked down CBP beta, we actually see a reduction overall. So we compared these two and looked to see what were in common, and in particular focused on the cytokines that were oppositely regulated in the two models, and identified 16 different proteins. Obviously, we cross-referenced this to human tumors and so on, and of course they're expressed by some tumors and not others. There's a lot of variability there, but we did take 3 that were commonly expressed in human cancers and tested them for their ability to, of course, drive myotube atrophy in culture. So these are the Serpent F1, the TNFRSF11B, and CD39, and all of them can cause wasting when added to culture. So, I mean, this is sort of the endpoint of this project, but it opened up a lot of other questions for us. In particular, where do you go from here? Well, I mean, for me, the idea that the tumor secretome could communicate with muscle satellite cells, and this changed their secretome into something that was procachectic, I found, particularly interesting, that there was this connection between a highly variable tumor secretome and then a response that seemed to be in common in the muscle satellite cells, and that secretome itself could lead to wasting, or also the impairment of differentiation. So we really wanted to sort of understand now how highly variable secretomes in the human cancers can somehow elicit the same response in muscle, right? So impair regeneration and result in muscle damage. So we tried to look at this in a more CBP beta biased way to see whether or not there was any sort of tissue restriction or anything that was in common. So I'm really interested in understanding whether or not other cells in the muscle microenvironment actually can respond to the tumor secretome the way that muscle satellite cells do. And I'm obviously very curious about what the signaling pathways are that actually turn on CBP beta expression in muscle satellite cells. Certainly in the context of therapeutics, this becomes important. So we actually just used a single cell RNA-seq database for muscle to look at the sort of the human cancer secretome in general, and started to look at where the receptors for those secreted proteins were. So we took 27 different human cancers, all of their secretomes, put them together, took only what we thought was CBP beta dependent, since that leads to cachexia, and then sort of mapped out where the receptors are. And I think what you can see really quickly is that at the top two lines here, these are myonuclei. So this is actually the myofibers. And the myofibers don't really seem to be the primary target of the tumor secretome itself. In fact, we can see that myoblasts and muscle satellite cells do have some of the receptors that are expressed on them. But if you look at the clusters in particular, it's obvious that FAPs are highly responsive to what is in that tumor secretome, as are, so these are the FAPs up here, as are cells in the monocyte and macrophage lineage. And so this is obviously an area that we find really interesting, because it does suggest that these populations are actually really important in detecting that signal that's coming from the tumor, and may be important in terms of, depending on what their reaction is to that signal, they could be contributing directly to the pathogenesis of cancer cachexia. And that's something that certainly my lab is very interested in. Obviously, CBP beta expression, if you look at muscle, just whole muscle, from mouse models of cancer cachexia, so the LLC model I showed you, the C26 model of colon cancer and pancreatic cancer, but as well in human cancers, CBP beta level is really, really highly expressed. And this is leading to dysfunction in the muscle microenvironment itself, including increases in CD31 staining, so more endothelial cells that are present, and also changes in macrophage polarization. So we're certainly seeing far more M1 type macrophages in the cachectic environment than in a healthy muscle. And in fact, even the production of some of these key cytokines that we know are bad for muscle, like IL-6 and IL-1 beta in the context of cachexia, they're not necessarily being expressed by the myofibers themselves, but seem to be expressed strongly by cells like FAPs and macrophages. And so we really are interested in understanding how the tumor microenvironment also responds not the tumor microenvironment, the muscle microenvironment responds to the tumor itself, and what the consequences of that would be. So what I've shown you is data of, what happens to the satellite cells in the context of cachexia. Certainly where we're going now, our next step was looking at what happens to the myofiber. So if you take myotubes and you incubate them with the conditioned media, does their secretome change? And the answer is absolutely, it most certainly does. And what we see is upregulation, again, of a lot of pro-inflammatory cytokines, suggesting that the myofibers themselves are also actively secreting compounds. Most interesting is some of the most strongly upregulated compounds are chemotactic factors, which could be involved in the recruitment of immune cells to the muscle itself. And that can have an impact not only on damage in the environment, but also polarization of macrophages and so on. So there's a lot that's going on in this muscle, but we don't really necessarily understand what happens first. And I think really where we're going now is trying to break it down very early of what are the first initiating events, which cells respond first, what do they do, and how does that change, leading us all the way to the myofiber itself and that wasting. And I think that there's potential here for us to also imagine that as this inflammatory signal spreads in skeletal muscle, because of its overall mass, we're actually increasing inflammation overall in the patient themselves. I guess the last little bit, and this is just my last slide, was trying to understand what regulates CBP beta expression. And this has been kind of a big black box overall in the field for anybody who works with this transcription factor. It's known that it gets upregulated downstream of cyclic AMP signaling, but realistically, other than that, we didn't know. But we thought it would be interesting to try and identify approved drugs that could potentially upregulate or downregulate CBP beta protein expression, because we thought this could have some therapeutic benefit. So this is just some preliminary data from a drug screen that we've completed, and what you can see is that many of our tested compounds actually upregulate CBP beta expression, and so this could have an impact, obviously, on muscle quality in those. But there are also numerous compounds that can downregulate CBP beta expression. What they have in common is often GPCR signaling. And so really what we're looking at is downstream signaling from GPCRs as being sort of the many roads that lead to, of course, changes in CBP beta expression. And so this might be one of the keys that allows us to understand how a really variable tumor secretome can actually, despite the fact that it's quite variable in all these different cancers, lead to very, very tight biological responses at the level of the muscle, so that many roads are leading to basically one or a few cellular responses, and in that are leading, of course, to wasting in the context of cachexia. So I will, of course, do my acknowledgements of all of the people who have done the work. And so this is my lab right now. Most of the work that I showed you is from a former PhD student, Hamoud El-Soudes, who did most of the work looking at beta expression in the tumor and its contribution to cachexia. Much of the bioinformatics work was done by a master's student named Isra Ahmad. All of the work, starting with CBP beta expression, were done by François Marchildon and Nina. Current work that I showed you that we're working on right now was done by Natasha Strong, Alex Brown, and Aisha Saleh. So obviously, I want to thank all of my funders as well, and thank you very much for your attention, and I will do my best to answer any questions you might have.
Wow, Nadine, thank you very much. What a compelling story. It's a great story. Such good evidence you provide. That's great. So folks, let's take some questions through the Q&A opportunity there. If you don't mind, type your answer, your question in there for Nadine, and we'll take a few questions before we take a break for the weekend. So just let me start by asking, I need some clarity on the expression of CBP beta in muscle. Mm-hmm. You just showed a slide where, I guess it was in the tumor-bearing animals, where you saw high expression of CBP beta. But you said early on that it's really not expressed in muscle itself, it's expressed in satellite cells. I'm just, if I take a myoblast and do a Western blot, am I going to see any CBP beta?
Yeah.
But then if I take a myotube, I'm not going to see it. Is that correct?
No, you shouldn't. So there was one paper by a Japanese group that looked at CBP beta as being upstream of atrogen 1 expression. And so they saw, in the context of cachexia, what they thought was upregulation of CBP beta in the myofiber. Which was always interesting to us, because we thought, well, maybe that's a pathological response to the wasting itself. But we haven't been able to see that. Actually, none of our myofibers never seem to upregulate them, despite the fact that they're becoming pro-inflammatory. So this was very focused on CBP beta, but I think that there's inflammatory responses that are, of course, going through some of the other pathways that we've already established, like NF-kappa B and so on, that is contributing as well to the inflammation we see in muscle. So in muscle, healthy muscle, it's in the satellite cells, yep, quiescent and early activated, and then it disappears. It's not in the myofibers. We never really looked at that time at FAPS and other sort of muscle resident populations. So certainly that's where we're looking now, because there is some evidence, at least using the single cell RNA-seq data, that CBP beta is expressed in some of these other populations.
Okay. I'll just ask one more before we take a few Q&As here. I understand what you said at the beginning about a very small tumor can lead to, now I understand, because of this amplification that you just described. A very small tumor can lead to such big effects in terms of muscle wasting. And it seems like you provided a compelling story to help us understand that, because the tumors, secreting CBP beta that is then acting on cells that then secrete more cytokines that upregulate more of the CBP beta. Is that true?
Yeah, that's exactly it.
Okay. Okay, good. Okay, let's go to some questions. Anthony Chime, have you tried ex vivo experiments to more closely define the satellite cell action for activation, proliferation, differentiation with co-culture? Have you tried ex vivo experiments to more closely define satellite cell
So
action?
we've done ex vivo experiments to look at the role of CBP beta, particularly in sort of maintenance of quiescence and the self-renewal process. So sort of myofiber explants, culturing them, looking at PAX7, MyoD expression, and so on. We have not done it in the context of cachexia, though. So we haven't done those experiments. Which is actually really interesting to put the conditioned media on those. We've always just sort of done it with the myoblasts as opposed to on the fibers. But it's true that that particular experimental system would answer more specifically kind of where the cells get stuck. And that could be really, really interesting. But we suspect that they are proliferating. And I think Dennis Guttridge's lab showed that they were BRDU positive. So they are proliferating, but they just fail to differentiate.
Right. Okay. Nicholas Kaleo says, amazing talk. Could the overexpression of beta lead to a senescence-like phenotype in different cells in the muscle niche that could lead to muscle atrophy observed in a cachectic condition?
So that's a great question. And so, yeah, so Alex in my lab is very interested in looking at senescence as well. And so that the literature for CBP beta, because it's so broadly expressed, it's a little bit everywhere. And there's some evidence that CBP beta is involved in senescence in some tumor models, actually, like RAS-mediated oncogenesis. So there's potential there that in a cell type specific way that senescence might be contributing to this, and then therefore the senescence associated secretory phenotype would be involved in that as well. So I don't have any good evidence. It's one of those things that we think might be really interesting, but we don't actually have any evidence right now to be able to say conclusively that we're seeing senescence happening in the cachectic muscle.
Yeah. Okay. Okay. Let's see. Very interesting research. Chris Berry says, in the myoblast overexpressing beta experiments, was the cytokine release due to beta regulating their production or in response to some sort of cell damage in the models?
Yeah, so it's going to be the production, because most of our outputs in terms of measuring cytokines was looking either at RNA or the protein, but not secreted like from the whole cell extracts. So it's definitely production of it. And I guess in terms of our reductionist models, we don't have the innate immune system there to sort of respond to damage associated. That being said, that's a really interesting point, because I think for the myofiber, that could be really interesting, because what we're seeing is, of course, this sort of pro-inflammatory response and also these chemotactic factors that are being produced by the myofiber. And we know that the myofibers are being damaged in the context of cachexia. So it could be a damage signal like, hey, there's a mess here. We need help to clean it up. And that this is bringing in components of the innate immune system that can actually sort of compound the inflammation locally, right, and allow it to grow that way. And so that's definitely something that we're considering in the context of the myofiber itself.
Mm-hmm. Okay. Michaela Slavin asks about the heterodimer. What is CBP? What is beta heterodimerizing with to promote the expression of cytokines and subsequent inflammation?
So it looks like it's actually homodimerizing with itself. In that so there are several members of this family, and depending on what tissue you're in, there's different ones that are expressed. CBP beta is the predominant one in muscle, which has made things, I think, very easy for us to sort of assume that that's what the major pair is. That being said, as soon as you get into sort of pathology, then it's possible that, of course, other factors can associate. It is a BZIP transcription factor, so it can technically also heterodimerize with things like June and FOS, so other members of that larger superfamily, which would require a very careful analysis in terms of, sort of CHIP-seq type experiments to really identify what the different permutations are and pairs are that are leading to this secretome. Yeah.
Is ATF4 among those, or
Yep.
Yeah. Okay. Okay. I think we're out of questions. One last one I might ask is, I was interested in the variability among cancers in producing cachexia. So you mentioned early on that, of course, lung cancer, pancreatic cancer, very pro-cachectic kinds of situations. Are the different cancers proportional to the amount of, and their effects on cachexia proportional to this, to the amount of beta that they secrete? Do you think? Is that?
So beta has been linked with aggressiveness in general. And what we see by looking at these sort of large publicly available databases is that there is a correlation between the ones that are most cachectic and the highest levels of CBP beta expression. That being said, the human population, every tumor is unique. And so we've had the central idea is that cancers lose their tissue specific expression, right? They are cancers now. That's part of the criteria of it. But it still looks like in the secretome that they do maintain some level of tissue specificity. So it looks like, CBP beta in a lung tumor is expressing a group of genes that fall within what it can do in a lung cell versus what it can do in an ovarian cell versus what it can do in a pancreatic cell. And so that's one of the things that we're trying to understand is, okay, so it's expressing these collagens from one tumor, these collagens from another. How do we get from that to always the same effect in muscle?
Yeah. Fascinating topic. Okay. I think we better call it quits. That's a good set of questions and a great seminar. Stay with me, Nadine, please. Thank you all, folks, for coming and joining us today in the seminar series. That's the end of our talk for today. We'll see you at the next one. See you next time.
Thank you very much, everyone.